that will be able to stem those. I mean, we just know that, but she's so used to it that, you know, she looks at this and says, this is cancer without a doubt.
That's cancer. But it's not something you learn overnight. Got it. So here is an intermediate situation where you have a gland here, a gland here, a gland here, trying to be a gland there. The cells are of intermediate size. There are little dark spots that we call nucleoli that typically are not present in low-grade tumors and tend to be much bigger in high-grade tumors. So this is a pretty good example of an intermediate grade tumor. And there are no mitotic figures in this field. If we look around real carefully in other parts, we'll find some.
Maybe that one.
No, that's an apoptotic cell, I think. I wouldn't buy that. That's just a shortcut one. Next, you'll see a real mitotic figure. So this is a mitotic figure. Yeah, there's another one here. This elongated, almost worm-like thing is a mitotic figure inside a different part of the face. And these are bigger cells, and the nucleoli are more prominent. So this is, and there's only one gland structure here. So that's something that we would call high-grade.
And the nuclei are different sizes, they're different shapes.
That's true too. And also
there's a different amount of DNA in each one. There are different stages of the cell cycle. The nucleoli is where they make ribosomes, which are the machinery that makes protein. So there's bigger, juicier because they're proliferating. All of it's sort of related to each other. I mean,
the cell cycle is so broken in some of these cells that they go through S phase, but they don't divide. So you have
twice as many
twice
chromosomes,
as much, or three times, or 4 times as much.
In some cells and not in other cells. So it's that heterogeneity is like one of the things that Britt, without even thinking about it, sees and says, okay, that's a high-grade cancer. I mean, that's one of the things that you're looking at. In my mind, it's the genetics, which is underlying it all, and that creates what we're seeing in the microscope as this artifact that Britt described of, you know, this is what it looks like when you cut through it. But it's also related to the biology. And it's the biology that drives the invasion, the proliferation, the metastasis. So all those things are, you know, is what we're seeing all at once here. It's not just a pattern that is given a name. It's all related to the genetics and the biology. Sorry.
How common is to find necrosis of these cells in an aggressive tumor?
Oh, sure, it's common. You can see areas of necrosis, but it doesn't drive the grading, so it's not part of grading. But it's much more common to see necrosis in high-grade tumors than in low-grade tumors. So in pathology, and these kinds of gradings have been around much longer than we have known about the structure of DNA and the importance of all this. So empirically, pathologists have known that when the tumor looks like this, it's going to be more aggressive than the first one we saw. So they knew that before they know the reason for why they looks like this. So say you have that low-grade one, you get it at 1, 1, 1, you end up with 3 here. That would be extremely low-grade tumor. So 3 to 5 is a grade 1, 6 to 7 is a grade 2, and 8 to 9 score is a grade 3. So that's what you would see in a pathology report that you give it, and it's called a Bloom-Richardson type score system that most everybody now uses. Tumor stage. Have you guys heard about tumor stage? Yeah, I thought so. So we can skip that part because we have so little time. Okay.
Just get it all down.
Yeah, get it all down. So I'm going to go through this pretty quickly. So they're lobular carcinoma, and tubular, and mucinous, and medullary, and some other special types. And I'm not going to go through and expect you to know this. I'm just going to show you a couple of different patterns so you get a feel for it. So next. So this is what you have seen already, several examples of an invasive ductal carcinoma, garden variety, intermediate grade. And next, here is what typically lobular carcinoma does. Instead of growing in clumps and glands, it grows in individual strands like this. It has this peculiar invasive pattern of single boxcar, some people call it, arrangement. Very classic for lobular. And the cells are very small. And they can be tricky to recognize because the cells look very much like lymphocytes, and lymphocytes also can come in little strings like that. And lobular carcinoma is not necessarily one big area of tumor, it can be little pinheads scattered throughout the breast tissue. And it can be actually very tricky to recognize that there is widespread cancer in the breast if you don't pay very close attention to those little small areas of single row cells. And sometimes it's hard even when you think about it, so you can do a stain that distinguishes between lymphocytes and epithelial cells, and then call it keratin stain, and that can be very helpful. Next.
So in that case, the nuclear morphology is not indicative?
Oh, it is. Lobular carcinoma is typically much smaller cells than intermediate grade ductal carcinoma. However, low grade ductal carcinoma and lobular, the nuclei can look very similar. But on the average, if you take all ductal cancers and compare it to lobular cancer, ductal cancers have bigger cells. And they grow in this different pattern. So sometimes, if you're not sure, you can see also a mixture of the two patterns, and it's not always so clear what's going on. You can do this e-coherence stain that I already mentioned, which will stain a membranous pattern. You can see sort of a lace-like pattern here around the cells in a ductal carcinoma. So that can be very helpful.
But the lobular, by
Lobular is
definition,
negative.
doesn't have the E-cadherin.
Right.
And in fact, I'll get to it in a second, but E-cadherin is on the surface, and it is what sort of glues cells together to each other, right? It's a cell-cell interaction protein. And so if it's gone, then they're no longer in these clusters and these ductular type, things, but instead they're all separate and sort of
And
growing,
they're sort of crawling along the stroma.
right, crawling in through a crack in the stroma. But because they don't have the cadherin. And that's why it grows in that pattern.
So it's a very useful tool for a pathologist to be able to tell the two.
And it used to be that it would be used to confirm a lobular. Now it's used in order to define what's lobular and what isn't.
Right.
It has to be E-cadherin negative.
Right.
It used to be there were a few percent that were E-cadherin.
You can still
But no
argue, but
longer.
we're not going to go into that.
Okay, so this is to explain that if you look at the genetics of these cancers, and this is a pattern that maybe you've seen, I'm not sure before, but when we do comparative genomic hybridization, which looks at the how many copies of each region of each chromosome, and like each box from, this is chromosome 1 and 2 and 3 all the way down to 22 and X here. At the end, when there's a green bar, it means something's gained relative to the rest, and a red bar means something's lost relative to the left. If you look at the top, that's 160 invasive ductal cancers. And so you see like a gain of 1 here. In fact, that dashed line is the P arm is the short arm of 1, and the long arm of 1 is the Q arm. That's the nomenclature we use for the structure of a chromosome. So you can see 1Q is gained, and 8Q is gained, and 8P is lost, and so forth. There's chromosomal changes that are typical of these breast cancers. And here's the lobular breast cancers, different from the ductal ones that Britt was just describing. Look, it also has the 1Q gains and the 1P losses and the 8Q gains and the 8P losses. So a lot of exactly the same thing. So just because they look different doesn't mean that the genetic changes are necessarily different, except down here, 16Q. This chromosome arm is lost in almost 100% of the invasive lobular cancers, and in only 30 or 40% of the invasive ductal cancers. So what's going on 16Q? Anybody want to guess what protein is located on chromosome 16Q? The long arm? E-cadherin, exactly. So E-cadherin maybe is mutated on one, because we all have 2 chromosomes of every type, right? One from mom, one from dad. So one of them is lost in all the lobular cancers, and the other one is probably mutated. And so you don't have E-cadherin, which is why lobular cancer looks like lobular cancer and behaves like lobular cancer. So that's the theme again, that the pathology is telling us, showing us a pattern that really represents the genetics and the biology the same time.